Good morning, students. In this, I am going to explain about plant viruses. Plant viruses, you have three, that is TMV in detail, the transmission and effects, and you have other two viruses, the slow viruses and DI viruses, that you have to study only the important discovery, how did they discover, and the importance. So, in this lecture, I am going to talk about only TMV. So TMB was like the first few viruses to be studied because the Chamberlain filters, with using that which Dmitry Ivanovsky, which I was talking in under classification of viruses, where he found that after filtering the sap through Chamberlain filters, it was still infective. So later on, people started to isolate the TMV virus, and it was Stanley in 1935 who crystallized the virus for the first time, for which he was part of the Nobel Prize winner. And then further, it was like electron micrograph pictures were taken, and then it was identified. It is consists of nucleic acid called RNA, and it has a protein coat. Like any other viruses, these are also obligate intracellular parasites. Outside, they stay as inactive particles, and when they are inside the cells, they become active, and then they replicate and then propagate from one cell to another. TMV belongs to the class Alsoviricites, order Martilivirales, family Bircaviridae, genus Tobamovirus, and species Tobacco mosaic virus. This virus infects the family Solanaceae. This virus has a characteristic mosaic-like structure, that is patches of decoloration, and then you can see that the virus is infecting the whole plant easily. So morphology, again, it has the capsomeres, the coat protein, and then it has the internal RNA. So tobacco mosaic virus is lord-like appearance. Its capsid is made up of 2130 molecules of coat proteins and one molecule of genomic single-strand RNA. The length of the RNA is 6400 bases long. The coat protein self-assembles into the long rod-like helical structure around the RNA, which forms like this RNA has hairpin loops where they attract this coat protein and they stabilize and then form these loops. The protein monomer consists of 158 amino acids which are assembled into four main alpha helices which are joined by prominent loop proximal to the axis of virion. These virions are 300 nanometer in length and 18 nanometer in diameter. Negatively stained electron microscope picture, you can see a nice channel with a radius of 2 nanometer. The RNA is located at the radius of 4 nanometer and is protected by the action of cellular enzymes by the coat protein. Okay, so the genome of the tobacco mosaic virus, I told it is 6400 bases long. It's a single stranded RNA. The 3' terminus has a tRNA like structure, and the 5' terminus has a methylated nucleotide cap, that is 7 methyl 3 phosphate of guanosine. The genome encodes 4 open reading frame. Okay, and these are like one is the capsid protein, other is the movement protein, and one is the replicase, which has the methyl transferase and the RNA helicase activity, that is an RNA dependent RNA polymerase, and another is a movement protein and a capsid protein. A single protein is synthesized, that is it's not named, but it has some function. So majorly 3 essential protein, which is the capsid protein, movement protein, and RNA dependent RNA polymerase. The transmission of this virus happens both mechanically, biologically, and physically. Physically, by wind blown, takes the virus particle and goes on to the other leaves. But mechanically, damage is very important for the spread of this virus. That is, contaminated hands causing scratches onto the leaf. That is the easiest way of entry, because it damages the cell wall, and then there are like space which is created through which the particle can easily enter. Or clothing or tools in the farm can be easy mode of transmission. Other than that, once it is inside the plant, the movement of the virus happens, and they are encoded within the seeds of the infected plants, or pollen grains, or insects which come and feed on the sap of the plant, are also easy transmitters of this virus. The multiplication cycle happens in 5 stages, that is, entry into the host cell, uncoating, intracellular development, and assembly, release. Entry into the host cell is basically by mechanical damage, either by the aphid making it damage, or by human intervention. There are no vectors for the uh, basically aphids can carry when they are taking the sap, because after cell to cell movement, the movement of to the different parts of the plant happens via the xylem and the phloem. So during that, aphids, when they feed on the phloem, they carry the virus from one plant to another. Once the virus has entered the cell by mechanical damage or pinocytosis, also there is uncovering or disassembly of the capsid protein. Now, this capsid protein is very interesting, because the virion has a highly stable capsid protein which is capable of surviving extended periods of time in the environment, that is up to 50 degrees. The dry leaf, the virion can survive, because there are very few vectors for this virus, so they survive in a thermostable manner. Upon cell entry, this stable virion must be destabilized to initiate virus translation and replication. This change in from stable to unstable is due to the presence of a stability switching mechanism that can respond to slight changes in the environment. It was found out that the mechanism of the switching could be provided by the repulsive interaction of carboxylate groups from glutamate and aspartate residues located at the interface of the capsid protein subunits. Like, if it is present in extracellular environment, these capsid proteins, they interact with the calcium ions or the protons, and they form a stable structure that their negative groups are not interacting, and destabilization does not occur. But once inside the cellular environment, because of the changes in the pH, or calcium ions get depleted, there is a loss of this protons or cations, allowing the closely located negatively charged carboxylate groups to interact, and thus the repulsive forces derived for interacting carboxylate group could then destabilize the virion and initiate disassembly. So there is structural deformation, and the RNA will be destabilized by this capsid proteins. They RNA binding capsid proteins therefore leave the RNA, and then this removal is from 5' end of the encapsulated RNA. So that is the uncoating mechanism of this virus. Once they are uncoated, there is biosynthesis of the viral RNA. I'll explain how biosynthesis happen in the next slide. The In order to produce disease, the virus must replicate and spread to neighboring cell and then systematically throughout the plant. Spread of the virus to neighboring cell occurs through a microscopic channel in the cell wall called plasmodesmata. These are slender structure extending from cell wall which connects adjacent plant cells. Now the spread from cell to cell, it is through xylem and phloem, that is systematic vasculature, to other parts of the plant. The cell to cell movement of the virus requires one or more protein called movement protein and other host protein. Most of the viruses uses capsid proteins for transfer. Now in this how this biosynthesis occurs, we'll study in two slides. That is, first slide I'll just tell how the RNAs are synthesized, and in the next slide I will tell how this movement happen from the attachment site to plasmodesmata. The whole cell is made up of actin networks. How this channeling happens through the microtubules and then crossing the plasmodesmata. First, viral RNA replication. It is made up of single strand positive sense RNA as its genome. Replication of the virion, that is the RNA, through involves synthesis of the negative strand RNA using positive strand RNA as a template. The replication is at two steps: synthesis of negative strand RNA using positive strand RNA as a template, which forms the double stranded intermediate termed as a replicative form. Now, the next is the synthesis of positive strand RNA using negative strand RNA as a template, and then that is encoded, virus encoded RNA dependent RNA polymerase. Now you have the conversion of the positive strand to negative strand. The RNA dependent RNA polymerase has helicase activity and replicative activity, which creates the negative strand, and from that you have multiple RNAs coming out, which is helping the synthesis of capsid monomers and then other movement proteins, what is required by the TMP. So how this happen transfer? Once you just concentrate on the small letters like EA is at the site of entry below. TMP RNA is released. Okay, the site of entry, how does it happen? Through opening in the cell wall by mechanical damage. The cell wall and plasma membrane forms the opening through which the TMP can enter, or through pinocytosis after wounding. The TMP RNA, the B, you have to check now, this one, B. Once the TMP is inside the cell through wound or pinocytosis, the TMP RNA is released from the capsid at the site of viral RNA, and the viral RNA granule formation happens. The granules are associated with the endoplasmic reticulum, which may serve as a replication site on transport of viral RNA to cortical vertices or perinuclear regions of the endoplasmic reticulum. Transport to these locations requires microfilament, that is MF. Okay, transport to the locations. Other membrane such as a vascular membrane may serve as a scaffold for viral replication. A viral replication complex, that is VRC, is formed in the cortex vertices or perinuclear regions of the endoplasmic reticulum. This one, VRC. VRC contain viral RNA, movement protein, replication protein of the host, that is TOM1. TOM1 is a membrane protein and interacts with the replication protein and serves as the anchor between the replication protein and the host membrane. So people think that this TOM1 can be derived from ER or vacuole or any other membrane. For TMB intracellular movement, this virus replication complex move from the site of the replication to plasmodesmata During that, elongation factor 1A interacts with viral RNA replication, microtubules, and the microfilaments, and influence TMP movement. An interaction between two host protein, that is class 2 not at 1 like protein, that is NTH201, and DNA J like protein MP1P1, MPIP1, and the TMB uh, aid in the transport of the virus to plasmodesmata. The influence of the microtubule end protein, that is EB1A, on the virus is placed during the transport to plasmodesmata. Moment protein microfilament serving activity at the plasmodesmata is proposed to eliminate F-actin like structures at the plasmodesmata to increase the plasmodesmata size exclusion limit, that is SCL. In addition to interaction with movement protein, the ankyrin repeat protein ANK is correlated with the increase in the size of plasmodesmata. Viral RNA transport through plasmodesmata within the viral replication complex, or simply with viral RNA and movement protein. The movement proteins are phosphorylated in the plasmodesmata to release the viral RNA from the translation in the next cell. After the viral RNA transfer to the neighboring cell, viral replication complex remnants associate with endosomes, okay, for transport to vacuoles through interaction with vesicle fusing protein synaptotagmin with movement protein. Transport is proposed that the actomyosin network is involved. So okay, this is how from one cell to other cell the transport happens. Once the other cell receive this viral replication complex, they also synthesize subcellular mRNAs or genomic RNAs via the same mechanism, and then transport from one cell to other cell. Now, once the subcellular RNA synthesize the capsid protein, the maturation occurs. It's a very organized process. Initially, the capsid assembly begins with the capsomeres associated with 3' end of the RNA. TMB assembly will occur in vitro when the capsid proteins and nucleic acid are mixed. So, how this occurs? And this the assembly is by the formation of 20S complex. The monomers, they form dimers or trimers or 4S A protein, and then they form 20S complex, that is disc-like complex. This 20S aggregates are required for efficient nucleation with viral RNA, and the viral RNA by virion assembly initiates at the RNA origin of assembly, that is OAS. A stem loop structure and around 876 nucleotides from the 3' end of the RNA is formed. This stem loop like structure with the capsid proteins is what the like the stem loop like structure is what attracts the capsid protein, and then one more stem loop like structure is formed, then attracts the other capsid protein, and that is the 20S aggregates. In this way, more and more 20S aggregates are attracted towards the mRNA, and the virion elongates. Elongation occurs in both the directions, that is 5' and 3' direction of the RNA. Elongation in 5' direction occurs by an inside out mechanism. In the 5' end, the RNA is pulled up through the central hole of the helix. Um and then in the 5' direction, the RNA and capsid protein assembly is very fast, and aggregation of this 20s aggregates is like very fastly assembles. Assembly at the 3' direction is considerably slow and is thought to involve is addition of single subunits or A protein aggregates, that is the trimers or dimers, okay, which is the reason it is very small. At neutral pH, the carboxylic acid of the adjacent capsomers have normal pKa values, but at the acidic pH, the repulsion between the adjacent carboxylic group occur due to increase in pKa. This results in lock, that is washer formation, and there is aggregation of this capsid protomers. Once the aggregation happens, the loops which are formed by the RNA, that is OAS, that is RNA origin of assembly, attracts this 20S monomers, and then assembly moves from 5' direction and 3' direction both. The release of the plant viruses is by lysis of the plant cell. Once enough of the viral particles are made within the host cell, there is disruption of the host cell, and then there is release of the host. The release of the virus. The cycle of TMB involves attachment. The attachment is by mechanical damage and getting into the cell or pinocytosis, or through the aphids. And then once inside the cell, the virion RNA is disassembled from the capsid monomers, and the RNA gets into the nucleus. Once inside the nucleus, it synthesizes the mRNAs required, and the genomic RNA, and that is single-stranded positive sense RNA, that is accumulated in the cytoplasm, and then you have the capsid monomers are also synthesized, that is assembled as a virion, and once the virions are assembled, they are ready to be lysed from the cells and released. So this is the cycle of TMB.